Hello everyone, I'm Cassandra Joyce or Alphonse, and I will be discussing different techniques in studying plant cells and tissues. Let us start. So the first technique is called the dry mounting. It requires a small or thin section of the specimen and is directly mounted on the top of the slide. So I have here my slide, and the specimen that I will be using is an anther. Place the specimen on the center of the slide and place the cover slip directly above the specimen, and your slide is now ready to be observed under the microscope. The second technique is called the wet mounting. In the wet mounting, specimens are suspended in liquid medium but not in a resinous substances. So there is my second slide, and the specimen that I will be using is a cutout letter E from a magazine. First is leave a drop of water on the center of the slide, and then place your specimen or the letter E on the center or on the top of the water drop. And then at an angle of 45 degrees, place one side of the cover slip against the slide, making contact with the outer edge of the liquid drop. 45 degrees. Then release the slide. It is now ready to be observed under the microscope. The third technique is called the freehand technique. In this technique, place your forearms resting on the table and your elbows. Hold the specimen between your thumb and your index finger. I am using a stem. Section slowly by drawing the razor blade toward you in a slicing motion. Transfer sections to a Petri plate containing 0.05 toluidine blue. In this video, I am using a water instead of a toluidine blue. Mount sections in a clean slides in a drop of water. There is our specimen. And then cover it. Lower the cover slip on the specimen. And then now observe the slide under the microscope. The technique is called the squash technique. It involves the spreading of the cell masses into a single layer of cells. I have here my slide, and then I will be placing a plant material or a banana in the center of the slide. Okay. And I will be placing another slide on top of the specimen, then gently press it. Press the upper slide gently with an eraser of the pencil to make it even spread. After pulling the slides apart, put 2 to 3 drops of water on the specimen, then place your cover slip You can use a low power objective to look for areas where the cells have been squashed and spread apart in such a way that there are few or no overlapping cells. The fifth technique is called the epidermal peel technique that is ideal for preparing dermal tissues of any laminar plant organ. I have here a half cut of a leaf. Tear the laminar plant organ in a twisting motion so that the epidermis is peeled away. Whenever I try to cut the leaf in the twisting motion, my leaf turns out to be like this. So I prepared a part of the leaf where this side was already peeled that can be used for the observation. The next step to do is to continue the wet mounting technique. Diba, drop on the slide. Then place our specimen on the top of the water drop. Then at an angle of 45 degrees, cover the specimen by using the cover slip. And it is now ready to be observed under the microscope. The last technique is called the sectioning. Specimens for wet mounting or even for permanent mounting are cut in different ways or planes depending on what is needed to be observed. The first type of sectioning is called the cross section or the transverse section. The cut is done horizontally or laterally. The second type of sectioning is called the longitudinal section, where the cut is done vertically or longitudinally. The first type of longitudinal section is the tangential longitudinal section. The cut is done tangent to the preference or vertically.
Happy Daddy! Happy Daddy! Happy Daddy! To you!
The second type is called the radial longitudinal section, where the cut is done at the center, vertically. And those are the techniques in studying plant cells and tissues. That is all. Thank you.